intestinal tract of humans and warm-blooded animals, where it exerts actually a number of beneficial functions. Nevertheless, in certain strains of E. coli actually develop the capability to cause disease in human and animals, mainly in the gastrointestinal environment, which is the natural site of these microorganisms, but also they have evolved the ability to cause disease in generally in all body compartments, including the kidney, the urinary tract, and all the other organs through bacteremia. This is because this particular species has a huge genomic plasticity that actually make it able to adapt to almost any kind of niche. My presentation will mostly deal with diarrheagenic E. coli, which are all these E. coli strains that have evolved the capability to cause gastrointestinal diseases in humans. And this particular class can be broken down into pathogenic groups, or pathogroups or pathotypes, that can be distinguished by the ability they do have of colonizing the intestinal mucosas in different manners, or the ability to produce peculiar toxins. And as a whole, we have at least 6 different pathogroups known as enteropathogenic E. coli, Shiga toxin-producing E. coli, and particularly a subgroup known as enterohemorrhagic E. coli, which cause very severe diseases in humans, and enterotoxigenic, and there are enteroaggregative, enteroinvasive, and diffusely adherent E. coli groups. All these pathogroups of E. coli have peculiar epidemiological roots. Most of the groups are diffused through an inter-human transmission cycles, mostly involving oral fecal routes. And as a matter of fact, they are leading causing and the most abundant pathogens in developing countries, causing a huge number of outbreaks of infection characterized by high mortality rates. This is true for most of the diarrheagenic E. coli group, which include enterotoxigenic E. coli, enteroinvasive, enteroaggregative E. coli, and enteropathogenic E. coli, while on the other side, there are the Shiga toxin-producing E. coli, which have a peculiar epidemiology. In fact, they are zoonotic pathogens. That means that they have an animal reservoir, which is the gastrointestinal tract of ruminants, and infection are mainly transmitted to the consumption of contaminated food, but also through a number of ways environmentally connected. Let's say direct contact with the environment, particularly water, which might become contaminated through contact with feces from ruminant, generally from the zootechnic activities, or by consumption of vegetables, which have been grown in crops irrigated with contaminated waters or fertilized with not properly matured animal manure. Well, I meant to say that actually the rigid categorization into pathogroups sometimes doesn't work in a so precise way. This case is well illustrated by what happened, let's say 3 years ago, during a very large outbreak of Shiga toxin-producing E. coli infection that occurred in Germany and in France during the May-June 2011. That episode was indeed one of the largest outbreak of staph infection, and for sure the most severe one ever recorded in Europe. As a matter of fact, we had some 4,000 cases of infection, with some 900 severe cases that required hospitalization and dialysis treatment, and some 50 deaths. In that case, the strain was somehow set transversally to the groups that I've been showing to you, because it was actually an enteroaggregative E. coli that had acquired the capability to produce Shiga toxin through the acquisition of a bacteriophage conveying the genes encoding the Shiga toxin themselves. And this hybrid, let's say, virulence feature conferred to this strain a particular virulence. It was really, really aggressive, as it was witnessed by the high number of cases. and by the number of severe cases. Immediately after its appearance, the scientific community started debating on which could have been the setting that may have furthered the emergence of such a pathogenic strain, because in order to have the emergence of a strain with the features of a human strain mixed to feature of other E. coli present in an animal reservoir, of course, the two reservoirs must have been in contact at a certain moment. And for the shigotoxin converting phages to jump from one human-born E. coli to a zoonotic one. And therefore, we decided to make a piece of work in that direction. And following to the collaboration we had with this university in Nigeria, we carried out this piece of work somewhere here at the end of the red arrow, particularly in the area of Zaria city in Nigeria, where actually a very appealing from the microbiological point of view setting was available to us, with a very large abattoir, slaughtering some 15,000 animals per year, that used to collect the effluents. The effluents are, the mix of the, animal feces, water, blood, and other substances of discharge of the slaughtering process, collected into a tank, which spills into a small water stream, flowing quite aside to the abattoir. In turn, the water stream flowed into a small river, Korai River, whose water was used to irrigate the crops grown nearby. So this was a very peculiar setting, and indeed we decided to perform a sampling scheme right there with a number of samples taken from the feces of the animal just before slaughtering, but also from the effluents, both in the tank or in the small water stream, and samples taken at the river, and finally samples taken at the at the farms where the vegetables were growing. And actually we had to divide the activity in two parts, because Nigeria is not an easy place to go or to transfer material from to Rome here, so all the microbiological part of the work was done there directly on site, and basically we decided to keep the work as simple as possible because of the lack of many facilities there, such as the PCR facilities, which are not available on site. So just a microbiological examination with E. coli isolation, and decided to move for an enzyme immunoassay for assessing the capability of the isolated E. coli strain to produce shigotoxin. Once a number of strains have been isolated and collected, they have shipped to Rome for the confirmation of the shigotoxin production by using cell culture assays and the viral typing by PCR screening for the virulence genes identifying all the virogenic E. coli strain I've been talking about. Let's come to a few results. As a whole, we assayed more than 700 samples, including those from the abattoir, the effluent, the water stream, the river, and the vegetables, and could isolate biochemically confirmed E. coli from 520 of them. All these samples have been screened with the enzyme immunoassay, and 43 colonies resulted to be E. coli and able to produce shigotoxin, at least enzymatically, let's say, by immunoassay. Then the strain had been sent here to Rome, but unfortunately, due to difficulties in getting the permission to move biological substances from Nigeria to Italy and so, the strain stayed there for 8 months, and a few of them actually died. And when they arrived here, we assayed them and could confirm the production of virucytid of shigotoxin from 8 of them. So the same 37 strains were subjected to viral typing by PCR and assayed genes, the genes encoding the Shiga toxin and a number of intimine, adhesin, and other colonization connected factors specific for the other dirogenic E. coli were also included in the screening, as well as the subtilase cytotoxin, which is a recently described toxin quite close to the Shiga toxin, but having a different tropism within the cell. And we found some interesting, although the majority of strains seem to be, let's say, fake from the perspective of producing Shiga toxin, which might not be true because I said they have been kept for 8 months, in not really favorable conditions, and Shiga toxin coding genes are conveyed by bacteriophages, so they might have been lost during the storage. But anyway, what's interesting is that actually 3 strains gave a positive cytopathic effect on virus cells, which was supposed to be Shiga toxin, but were negative for the Shiga toxin coding gene, so they could, produce another kind of toxin. We are still working on that. But what is interesting is that we had strains with this peculiar profile from the fecal content of the animal at the slaughterhouse and from one of the cabbage samples taken at the farms. More example of possible connection between the abattoir and the crops in the field were made up by other strains which were more or less typically Shiga toxin producing E. coli, again from the fecal content and the cabbage, and 2 subtilase producing strains which again were present both in the effluent, that means that were in the animals, most probably, and crops irrigated with the water of the Correy River. And last but not least, we could isolate an enteraggregative E. coli there. So actually this is the first evidence of the contemporary presence of an enteraggregative E. coli together with Shiga toxin producing E. coli in the same environment. And actually, of course, we don't have striking evidences, but we can hypothesize that in such a setting, the emergence of the new group of enteraggregative E. coli producing Shiga toxin might have occurred. So, summing up, I can say that according to our results, of course, in low hygienic condition, the abattoirs operation might spread pathogens in the environment, and this is quite obvious, but I have to say that in developing countries, such kind of low hygienic conditions are very common. So you can figure out what kind of pressure the environment is subjected to when it's close to the environment operation. That the spreading of pathogens might involve both human-borne and zoonotic pathogens, favoring a reshuffling of virulence determinants. And finally, I can say that we actually could have evidences of the transfer of pathogenic E. coli strains from the animals at the slaughterhouse and the crops irrigated with the fields where the effluents from the abattoir were collected. Just before closing, I would like to acknowledge my colleague at the University of Zaria and the ISS right here, and thank you very much for your attention.
Time for maybe one very short question.
Or maybe not. Oh.
Thank you.
Are quorum sensors involved in the manifestation of the toxicity?
Oh. Okay, if I got it correctly, you are asking, if quarine sensing is involved in the manifestation of toxicity. Well, actually is very much involved. And indeed, either the production of Shiga toxin, but also the coordinated regulation of the other virulence genes present in the Shiga toxin producing E. coli genomic scaffold, it is finally regulated by quorum sensing. And indeed, there are actually a number of papers already published which are not completely conclusive, but displaying striking evidence that the quorum sensing molecules can trigger the production of the toxin and can modulate the regulation of other virulence determinants in this pathogenic bacteria. My pleasure.
Okay, so thank you very
Thank you very
much.
much.
Let's go. Let's go on to the next speaker.